Hi there, I'm Kate, a scientific content writer at Izon Science. In our previous episodes, we've explored how extracellular vesicles, or EVs, can be isolated from urine and milk. Today, we'll dive into the world of cerebrospinal fluid. Cerebrospinal fluid, or CSF for short, is that clear fluid that surrounds your brain and spinal cord. It's like a protective cushion that helps keep your nervous system safe, while also playing critical roles in homeostasis, waste removal, and nutrient transport. Now, when it comes to sampling CSF, most of us would prefer to avoid a lumbar puncture, but for diagnosing central nervous system diseases and disorders, it can be essential. CSF serves as a window to the central nervous system, and EVs could be vital in this context. EVs contain proteins, nucleic acids, and other small molecules, all encapsulated in a lipid bilayer. These tiny, nano-sized packages are secreted by all cells, and EVs derived from the CSF can carry biomarkers that help us understand what's happening inside the central nervous system. While small, EVs could be a big deal in brain research. Here's the thing. The concentration of EVs in CSF is much lower than that of plasma, about 5 times lower, and that's actually a good thing. It means that there will be less noise in the system from EVs derived from different cell types. This should help in identifying reliable biomarkers. One study found that 16% of CSF EVs are from neurons, with the remainder probably mostly from glial cells. Another advantage of CSF is that it has a much lower protein concentration compared to plasma, about 200 times lower. And that means protein contamination will be a far lesser problem. Plus, there are thousands of times less lipids in CSF, so lipoprotein contamination is also less of an issue. There are, though, of course, challenges with isolating EVs from CSF. Many EV researchers began by isolating EVs using ultracentrifugation. I know I did. There just weren't really any alternatives when I began my EV research. Many people still use this technique, but it just doesn't cut it when it comes to isolating EVs from CSF. But don't worry, we've got you covered with our QEV size exclusion chromatography columns. Not only do our columns isolate EVs from CSF with much greater efficiency than ultracentrifugation, but they also come in a range of sizes to fit your needs. Our 70 nanometer QEV columns have been shown to remove more free albumin contamination from CSF EV isolates than our 35 nanometer columns, or ultracentrifugation. And the best part? You can use them with virtually any volume of CSF without the need for sample dilution. That means you won't lose precious EVs in the process. Our QEV columns also have the advantage of being optimized to a range of input volumes from 150 microliters upwards to 100 ml, covering a huge range of CSF sample volumes. Now that you've discovered the power of QEV columns for isolating CSF EVs, you might be curious about the groundbreaking research being done in this field. Unfortunately, there is a catch. The quality of CSF EV research has fallen behind that of other biofluids. Many studies fail to isolate EVs, leading to complex samples that only contain a small fraction of EVs. Others have taken a DIY approach, ignoring established best practices for EV isolation. This lack of standardization makes interpreting results a tricky business, but progress is still being made in several areas of disease and disorder research. With caution and careful consideration, we can begin to unlock the mysteries of CSF EVs and their potential for revolutionizing the field of biomarker identification. Perhaps the most studied is Alzheimer's disease. Studies have revealed that EVs contain amyloid beta and tau proteins, which are hallmarks of the disease and possible contributors to pathology. Interestingly, EVs in Alzheimer's disease patients are found to have a lower concentration of these proteins, indicating that EVs might serve a protective role that is failing in the disease. Perhaps, as has been shown by another study, this may occur via the lower concentration of CSF EVs in Alzheimer's disease. However, the search for altered molecules in Alzheimer's disease, EVs such as proteins and microRNAs, has yet to yield any consistent results across studies. Unfortunately, the lack of standardized isolation techniques, or often any isolation techniques at all in CSF EV studies, has made it challenging to identify reliable biomarkers for any disease. This underscores the importance of standardized isolation methods and adherence to the MySafe guidelines in the field, both of which are vital for ensuring the reliability and reproducibility of research findings. With that in mind, where can the field go next? With the seriousness of central nervous system disorders and diseases, the need for early diagnosis is urgent. EVs could hold the key to this, but without standardized isolation and characterization methods between studies, more money will be wasted on projects destined not to succeed. Following the MySafe criteria would be a great benefit to the field. Another crucial aspect to consider is whether the benefits of CSF sampling outweigh the costs. While it's more invasive than blood sampling, if the biomarkers identified are significantly more effective than plasma biomarkers, it could be a game changer in early diagnosis and treatment. If these challenges can be overcome, the impact on the field of central nervous system disorders and diseases could be monumental. We have the potential to identify reliable biomarkers for early diagnosis and prognosis, allowing for more effective treatment options, but only with the right standardized methodology. It's up to the field to step up and meet these challenges head on. Only then can we unlock the full potential of CSF EVs as a diagnostic tool.